Forget frequently asked questions.
Common sense, common knowledge, or Google.
How about advice from a real genius? 95% of people in any profession are good enough to be qualified and licensed. 5%
go above and beyond.
They become very good at what they do, but only
0.1%
are real geniuses. Richard Jacobs has made it his life's mission to find them for you. He hunts down and interviews geniuses in every field: sleep science, cancer, stem cells, ketogenic diets, and more. Here come the geniuses. This is the Finding Genius Podcast
with Richard Jacobs.
Hello, this is Richard Jacobs with the Finding Genius Podcast. My guest today hails originally from Zimbabwe and then South Africa. His name is Clive Shiff. He's a professor now in the Department of Molecular Microbiology and Immunology at Johns Hopkins University. And he's been studying tropical parasitic diseases like Schistosomiasis, if I can pronounce that right, and Malaria, etc. So, Clive, thanks for coming. How are you doing?
It's a pleasure to be here.
Yeah. So, how long have you been studying infectious diseases, and what got you interested in them?
Well, I went to school in what was then Salisbury during the war, and we were a bit starved at the schools for good information because most of the teachers were in the army and stuff like that. So I didn't get much exposure to science. But when we actually started chemistry and physics, I began to realize that this is something that I want to do. And so when I finished at school, I went to South Africa to university in Natal, in Pietermaritzburg. And I took as my main subjects chemistry and zoology. And zoology introduced me to the world and the world of animals. And so at the back of my mind was, this is all exciting and great. You go down to the seaside and you can collect all these beautiful animals, strange things. But what the hell am I going to do back home? And it became clear to me that I had to specialize in entomology because there were no opportunities to work in science. And the main employer of scientists in the country at that time was the government. And so that's what you had to, sort of look at what sort of opportunities would arise. So I then left at the University of Natal and I went to Grahamstown, which is in the Eastern Cape. The university is called Rhodes University, and it is still called Rhodes University. And I was there a couple of months last year. As a matter of fact, I got an award from them. And
is that the same as the Rhodes Scholar? Is that where it comes from?
The Rhodes Scholar comes from Cecil Rhodes, and actually, he was. One can go into an interesting diversion here because he was a brilliant man, and he had great sort of projections of politics in the world. But actually, the headquarters of the Rhodes Scholarships is at Oxford. It's not in Africa at all. But anyway, I went to Rhodes University because they had a specialist course in entomology, and I did my master's in entomology. So when I came home, there were opportunities, and I started to work on. So in Zimbabwe at that time, there was a disease called sleeping sickness, which is
tsetse fly, right?
Yeah, tsetse fly. That's right. So I had learned entomology, so I joined the entomologists that were there, and the government was trying to eliminate tsetse fly, eliminate sleeping sickness, and the only way to do that at that stage was to get rid of the tsetse flies, because there's no drug that was effective for sleeping sickness. And the problem with sleeping sickness is that it also affects animals. I mean, there are several different parasites, so they're all transmitted the same way through the tsetse fly bite, and so we had to go out into the bush. And so I lived for 2 years in the bush with wild animals all around. And it was...
What's a couple of questions here. So what does sleeping sickness do to the person or the animal?
Well, it causes a debilitating disease. And it's in most cases, it's lethal. Some animals have now, wild animals are the host of the parasite, which are trypanosomes, and it has no effect on them at all because it's adapted to their physiology. But in domestic cattle, sheep, goats, dogs, cats, anything, in all mammals that have not been exposed to it are susceptible. And it causes, it doesn't put you to sleep. It causes a debilitating disease that is, the body wastes, and after about 6 months, you die. And
it's terrible.
It is. When I was there, they had a drug that they could use, but half the time it killed the patients. But if it didn't kill you, it would work once, and they wouldn't let you go back into the tsetse fly area again. Otherwise, if you got reinfected, that would be the end of the line. So,
is a tsetse fly administering a bacteria or a virus or a parasite? What is it?
It's a parasite. It's a trypanosome. The trypanosome parasites are extremely diverse. They can occur in plants because there are trypanosomes that live in plants, particularly plants that have got like rubbery sap, and it's the tsetse flies get infected by biting wild animals that have got the parasite, or of course animals that are going to die. And these tsetse flies live for about 9 months or so. They're very long-lived animals, and so rate of infection is high, and so they're extremely good in transferring the disease. But at that stage, there was a guy who joined the veterinary department who came from Cambridge in England as an entomologist. And he was working in the field, and he found that the tsetse flies are attracted very much to odor from cattle. And this is in the 1960s. I mean, people have got very sophisticated equipment now to look for what they call olfaction, attraction of hosts, of biting animals to the host. And he found that a simple chemical called octanol had attracted these tsetse flies. And you could make a trap with a gauze screen about 1 meter square and spray insecticide on that and take a little plastic, they sell for kids little tubes of colored water in plastic, and you freeze it and then suck it. They take the same plastic.
Oh, yeah, I remember those.
Yeah, and hang that up next to the cage. And these flies come in drove because they smell the octanol, and it's been successful. Tsetse flies are almost eliminated from Southern Africa. There's very few left.
Oh, wow.
In fact,
is there a tsetse fly repellent?
No, it's not a repellent. It's an attractant.
No, I know, but what if you had an attractant on one side of the house and then a repellent on the other side of the house, so that you'd attract and repel at the same time to different spots?
These things will bite you like hell. I mean, when they're hungry, you'd have to have a powerful repellent. The successful, and this is being used now for mosquitoes as well, to try and find what attracts mosquitoes to humans, because the mosquitoes are also transmitting malaria and other diseases. But it's much more sophisticated now. There are people that are dissecting into the brain of a mosquito to find out how the actual nerves are responding to these. What my friend Glyn Vale did, he dug a huge hole in the ground. When I say that, I mean big, about the size of an apartment. And they had cattle down there. And they had fans that blew the air from over the cattle and then out through tubes into the environment. And that the tsetse flies would come to those tubes. And this is how he worked it out. So really, that was interesting. I mean, I wasn't involved in that because after 2 years in the bush, I was either going to go nuts or should change into something else. And I started to work on other parasites because I joined the Ministry of Health then, and they were interested in other problems, you know. But always.
Would you mind talking about your time in the bush? I mean, how did you not get infected by a million parasites being out there, and what did you do out there?
Well, we lived in very Spartan quarters. They actually had aluminium houses, sheet aluminium. Here you call it aluminum, but in the rest of the world it's aluminium. But sheet that were all bolted together like an octagon and a roof, and you slept inside this. And when the temperature outside is very hot, you could cook inside these, they call it L10s. Yeah, but you had to be protected because there were leopards and lions in the area. I had a leopard run around my house trying to get in because my dog was inside with me and the leopard wanted a meal. So, I mean, the
dog must have been barking its head off, right?
No, the dog was barking its head off, yes. And then, of course, Jumbo, we have lots of elephants and rhino and things like that. I mean, I've had confrontations with all of these animals.
That's cool. That's amazing.
Walking through the bush, yes. You learn a lot, and it makes you feel very insecure.
Yeah, what did you notice about a leopard or a rhino or all these other animals? Like, you know, what did you feel like and what did you sense from them when you saw them? Like, were you truly in danger some of the times? And again, what did you sense when you were in the presence of these animals?
Well, the thing about the hippo is that they're not aggressive, but when they're frightened, they run. And the vegetation in areas where they live is very, very dense. So these animals can be a few yards away from you in a thicket that you can't see, and suddenly they will run out. And if you're in the way, they'll knock you down. I mean, they're not looking to attack you, but you've got to be nimble. This happened to me once too. Yeah. So I mean, it's a different world to live in. You're very much on your own. We were out in the bush for about 3 months at a time, and then we were allowed to come to a little town to get, food and groceries and things like that, and then we had to go back to camps because we were trying to control the tsetse flies, and that whole process morphed into using traps and chemical attractants, and that has worked. I mean, in that part of the world, there's malaria, and you have to worry about that. We took anti-malarials when I was in the bush, but that's not a way to solve the problem of malaria in the population. So the government had decided to control malaria, and in fact, some of the initial work in actually controlling malaria started in 1930. Well, there was an entomologist who came out to Rhodesia in 1932, and he stayed there for about 3 years studying the mosquitoes. And then he left a very detailed report on what he did. but there were no insecticides. The insecticides only came after the war, and the people in the Ministry of Health got hold of this special insecticide from the UK. No, it was from the US, and they called it DDT. And we found that you could spray it on the walls of a house, and the mosquitoes that transmit malaria tend to feed indoors. And when they've taken blood, they sit on the wall of the house or on curtains or something like that, so that they can digest the food and fly away. Because the mosquito is very small, and when it bites, it almost 4 times increased its weight 4 times. So
quadruple its weight.
Can't fly a long way. Yeah, so they fly onto the wall and sit there. And we sprayed the walls with insecticide once a year, and the incidence of malaria in the local hospital just dropped. So as a result, that was one of the observations that WHO was following up, because after that they decided to eradicate malaria, and that was now about 1955, 56, and that's when I joined them. It was interesting. I'd studied entomology, and I wanted to be an entomologist. And the WHO specialist who was seconded to the government said, there's no job for you as an entomologist, because we're going to get rid of malaria and you better find something else to work on. And so I did. I started to work on schistosomes, which is schistosomiasis is a disease. It's a parasite that lives in the blood. It's a worm. And these worms are about a centimeter long. They're not small. And they live in the blood vessels that drain the gut or that drain the bladder. And they are completely blind to the immune system. The immune system doesn't attack them. And these parasites can survive for years, 10, 15, 20 years in a person, and they have a lot of effect on the population. It's very difficult to study it, because we realized that kids were being affected, but it's very difficult to quantify the level of incapacity that a child has that's infected with this parasite. And the other thing is that if you find them infected with the parasite, you've got to do something about curing them. You can't just say, well, you've got this parasite and we want to test you. No, you have to cure them.
Well, what does schistosomiasis do?
Well, the parasites live in the blood vessels that drain the gut, and they lay eggs into the gut wall. And these eggs cause inflammation, and that allows them to get through the wall of the gut into the lumen of the gut where the food is, and they just get passed out in the excreta. And people who defecate in the river or near the river pass this infection. Well, they pass the eggs out. Now, the eggs, which are about 1/10 of a millimeter in length. So you can see them under a low-powered microscope, but you have to have a microscope. You can't see them normally. These eggs, when they go into water, they hatch and a larva comes out, and that larva swims around, and it can find certain species of aquatic snail, and they're very specific. And they invade the snail. And then inside the snail, they develop into a parasite that emerges after about 3 weeks. And this parasite emerges from the snail. The snails don't die. They just once they're infected for the rest of their life, which is about another 3 or 4 months. And
but then the snails can pass it to people?
And when you get in the water to swim, to bathe, or just to drink, these larvae go through your skin in a short period of time, because they secrete enzymes that can drill through the skin, and the larva then gets through into your blood, and it takes about 6 weeks to become an adult.
Okay.
And then it does the cycle again. And what we found, of course, over years is that children who are infected don't learn well at school. It's very debilitating. But if you treat the children and eliminate the parasites, then they go back to normal again. So kids used to do badly in school, and the parents would be complaining and take the child to the doctor, and the doctors there knew about bilharzia, about schistosomiasis, because it was a very common infection in kids. But it was very difficult to treat because the drugs were vicious drugs. I mean, they really made you sick. You had to, some of them you had to go into hospital for about a week and have a continuous injection of the drug.
I guess the good point is there's many potential checkpoints where you can interfere with schistosomiasis and its abilities. When it's in a snail, when it's in a person, before it's become an adult, etc. So, what therapies have been found to work with it?
So there were drugs which were based on antimony, and antimony is toxic like arsenic. So these were injected into people, antimony tartrate. That was when I started work. And then the pharmaceutical company Bayer in Germany found a drug that could be made into pills and used to treat people. And they had 2 or 3 of these different drugs over about a 10-year period. And in the 1970s, they came out with the final one, which was really very effective because it was innocuous. You could take it without getting sick, and it cured the infection. And that's what they use still now.
At what point in the infective cycle do you use it? And does it work at all points or only certain ones?
No, it kills the adult. It kills the adult. Yeah.
It kills the adult schistosomiasis or the adult?
Yes, the adult schistosomiasis, not the adult person.
Well, I wonder if there's any medicines like that. They only work in children, not in adults. I'm sure there is.
No, it works very well. Adults can take it. Children can take it. The problem is that, when these pharmaceutical companies design these drugs, they have a period of about 10 years when others are not allowed to make copies of them. But with this praziquantel, the drug that was made in Germany, in after about 10 years, it was made in many parts of the world, because schistosomiasis is in China, it's in the Philippines, it's in South America, it's all over Africa, and it really is debilitating. And what people have done, spent a lot of time and money is in trying to treat school children, mass drug administration. In fact, in Brazil, they decided that if more than 25% of the kids in a class were infected, they would just treat the whole class. They wouldn't even bother. And this is what they do now. People are asked, they have to sign an agreement form if you're going to do one of these mass drug administration courses, and then they treat everybody as in a short period of time. And of course, the doctors or the nurses or the clinical staff keep an eye on the ones that were treated, but they don't get sick from it. The drug is quite innocuous. But the problem, of course, is that this is curing your infection. And if you go back and sit in the water, you'll get reinfected. And this is what has happened. It's very difficult to eliminate this parasite. And people have been working, I worked on this all my life. And I remember when the Gates Foundation was very strong in supporting programs to treat school children all over the areas where this parasite occurs. And they they paid for it. And the research workers, mainly this work was done from people at the University of Georgia in Athens. They were the ones that got the money from Gates. And this was started about 2004, 2005. And here we are now, 2020. And the kids in the field are just the same infected because reinfection, the cure for schistosomiasis is improving sanitation. And this is what we did actually in Zimbabwe. We designed toilets that people could afford. If you design a pit latrine properly, there's no odor in it. There's no smell. And actually people use the toilets as a bathroom. So they'd go into a toilet with a bucket of water and defecate, then wash themselves, and the water all goes downhill. And the toilets don't fill up because the feces breaks down with bacterial effect, and it doesn't fill up. And the toilets are odorless. And this was done in Zimbabwe. It was done in Rhodesia still in the 1970s, and then it's all been published. But yet, I work in Zambia a lot now, and I can go into the field, and people build toilets the old way, just a hole in the ground, and there's no structure. There's no.
Why is that? Why is that?
Because it's all done, everything is done. I can be a little bit cynical. You see, it's not done by the government of Zambia or the government of Zimbabwe. It's done by people who come from overseas with good ideas. They get money and they say, we'll build a latrine. But they've never seen a latrine. All they know is that a latrine is a hole in the ground. And so they do this. And it's also, it's not only done by individual do-gooders, it's done by programs. UNICEF build toilets, but they don't make pit latrines that are odorless. If anybody's interested, all you have to do is to go to Google and say ventilated pit latrine, and you'll see the design. That's the design that we invented in Zimbabwe.
I wonder if even those porta potties in the U.S. are inferior to the design you're talking about.
Well, a porta potty has to be cleaned. The pit latrine doesn't.
That's what I mean. I said, I wonder even if the porta potties that they make in the U.S. or they use in the U.S. are not as good as these pit latrines you're describing. Sounds like it.
I don't think so. I don't think so. But the porta potties are expensive. These people, they don't have money to build a toilet that's going to be expensive. It has to be done with minimum expense, because most of the people living in rural areas live hand to mouth. They're peasants. They don't have a big income. So the design has to be very simple, and it has to be odorless. It has to be structured in an appropriate way. It can be built with mud and mud bricks. It doesn't have to be cement, and the only cement has to be for the floor of the latrine. And you need cement and reinforced rods. It's very easy to look at how these things were designed, because it's all over if you put the right keyword in, you'll see it in Google. I do that often just to remind myself. It's easy to get access to. So, I mean, the sad thing is we really haven't advanced in eliminating schistosomiasis from most of Africa. And it's also serious in China. The problem in China is that the parasite is a little bit more difficult to deal with. The snails are different, but in Africa and South America, mainly Brazil, the aquatic snails are there. And if somebody urinates into the water or defecates into the water, they get infected, and then the parasite will swim around, and anybody who goes in the water to swim or to bathe or whatever, wash their clothes, their mothers wash their babies in the water.
Yeah. So what role are you playing in all of this? How are you?
Well, what I'm trying to do now, really, because I go back to Africa. I have gone back frequently, mainly to Zambia and also Zimbabwe. But my real interest now is to improve diagnostics for these diseases, particularly schistosomiasis. But I'm working on a number of other parasites, because what we've found is that if you take urine from a person who's got schistosomiasis, and you filter that urine in the field through filter paper, cones that you had in class, the filter paper circles, you fold them so that it forms a cone, and you just pour the urine through that and let it drain out, and then dry the filter paper. The filter paper has to be dry, and you dry that. You can dry that in the sun, and then you put it in a Ziploc bag with a desiccant, and the DNA is stable. It'll last for years. And you take that to the lab, you can extract the DNA, and you can identify schistosome, hookworms, tapeworms, and now we found you can identify, there's a horrible tapeworm that occurs in dogs, which if humans ingest the eggs of the tapeworm, they get cysts inside their abdomen. Like, when I say a cyst, it's like a ball of tissue about the size of a tennis ball. And these things hang around inside your liver, your lungs, and there's no way to detect them. But we can find DNA from the parasite in their urine. And this is what I've been working on and still work on, and hope to publish a paper within the next month or so on this sort of work. Yeah, because this looking for DNA is much more sensitive. You get low-level infections.
Right. Well, very good. Well, Clive, you're really cool to talk to. There's so many awesome things that you've experienced, with animals and living in the bush and everything. But but for now, unfortunately, we're out of time. But okay. What's the best way for people to find out more about schistosomiasis and your work? Where can they go?
Well, Wikipedia is very good. Get good information from Wikipedia. And that's about the best place to get some. And the information I have is very accurate. So that's probably the easiest place to get. So if you just type in schistosomiasis on Wikipedia, you'll get 3 or 4 pages of stuff.
Okay. Okay. Well, very good. Well, Clive, thanks for coming. And it's great to talk to you. I appreciate it.
Yeah, it was a pleasure.
If you like this podcast, please click the link in the description to subscribe and review us on iTunes.
You've been listening to the Finding Genius podcast with Richard Jacobs. If you like what you hear, be sure to review and subscribe to the Finding Genius podcast on iTunes or wherever you listen to podcasts. And want to be smarter than everybody else? Become a premium member at findinggeniuspodcast.com. This podcast is for information only. No advice of any kind is being given. Any action you take or don't take as a result of listening is your sole responsibility. Consult professionals when advice is needed.